So we have forced all that stuff out. All 180 liters of that filtrate. Now I want to reabsorb it. Because ultimately I don't need to get rid of all of the things that get forced into the filtrate. I don't need to get rid of all of those ions, vitamins, minerals. The body needs some of that stuff, or potentially needs some of that stuff, right? So I want to be able to get that back into the body. That's where reabsorption comes in. As you can see from the picture here, there are a few different mechanisms through which we can transport stuff, solutes. We can do it through what's called paracellular. And paracellular is just, paracellular just movement in between, okay? There's a little bit of movement that occurs here in between the adjacent cells, okay? So let's say we do have some kind of inflammatory response going on here. Now you can see how we can get even more stuff reabsorbing or secreting, depending upon if we have some kind of inflammatory response occurring here. We can also do what's called transcellular, okay? And it just goes from one side and then across the other side, okay? One side of the membrane and the other side of the membrane, okay? From this side to this side. This is more like pinocytosis almost as well, okay? We can package it up into a little bitty vesicle and move it across. It can go through some specific protein channels. There are a multitude of ways that we can get this to go from one side of the cell to the next. Ultimately, it does have to be packaged up into a tiny little vesicle, and that's where that pinocytosis occurs, okay? Because remember, pinocytosis was that quote-unquote cell drinking, and this is basically kind of what it is, all right? This transcellular. So here is my glomerulus right here, okay? Here's my capsular space. And here is that filtrate entering into this proximal convoluted tubule, okay? And here's the wonderful microvilli appearance of that nonsense, okay? Oh, there's some other things kind of going on in here. Here's the peritubular capillary. It's this, okay? Here's the efferent arterial leading away. And then it helps to rise these little tiny little capillaries, blood vessels. This is the peritubular capillary. And then down here is my vasa recta. And this is that peritubular capillary that you're seeing running right along there, okay? And it's low hydrostatic pressure. Capillaries, by definition, are low pressure systems, right? But we have a high colloid osmotic pressure. It's because there's all the proteins are found here, right? An individual small amino acid not going to exert a whole hell of a lot of force compared to some of my large ones, which stay within the blood, right? So we have a high colloid osmotic pressure. So hopefully that just makes sense. Now, there were some things in the filtrate that get pushed out that the body is going to actively try to reabsorb, okay? If we look at one from a nutrient standpoint, there are two nutrients that we are getting pushed through that filtration membrane: glucose and amino acids. Now, under normal circumstances, those get pushed out, but the body wants to use that stuff, right? Glucose is going to be the primary provider of energy for the body. I don't really want to get rid of that. Again, I'm thinking under normal circumstances. I'm not thinking about somebody, who's a diabetic, who has an excessive amount of glucose in their blood, all right? When it's perfectly normal amount and you're not diabetic, all right? I'm not talking about those situations, okay? Under normal circumstances, all of that nutrient that gets thrown out, glucose, amino acids, they generally get reabsorbed at 100% because the body wants to utilize them. Glucose to make energy, proteins to make structures. When we look at how glucose gets absorbed, it is done through a symporter right here. To get glucose to come into the cell, it has to be paired with something. So we pair it with sodium. So it's a sodium glucose symporter or transporter, however you want to call it. The only way we get glucose to come across is when you pair it with sodium. It's the only way. Okay. So sodium goes from high to low, and that allows glucose entry into the cell. Otherwise, glucose wouldn't really want to go. Glucose is generally higher inside of a cell because that's where all the cells are going to be utilizing glucose. So to get glucose to go against that concentration gradient, we have to actively transport it. And we have to couple that with something. And we always couple it with sodium. So it's referred to as a glut transporter, G-U-L-T. And there are many types of glut transporters throughout the body. We have a glut transporter here in the kidneys. We have glut transporters in smooth muscle, glut transporters in the brain, glut transporters in skeletal muscle. We have a number of these glucose transporters throughout the different regions of the body here, okay? And it has to be active transport. It's referred to as secondary active transport because we pair it with a second substance. So there you go. But it's still going against its concentration gradient, hence active transport. Okay. Once we get it here, once we get glucose in, now we have a concentration gradient, right? High, relatively low here because we've just sucked it all out. And we can get glucose to easily go through this little port here on the basolateral surface and get it reabsorbed. And I can reabsorb all that glucose. Again, glucose is not normal in the urine, right? So if you do find it, that tells you you're having some issues going on with glucose in the body. Proteins are going to be the same way, okay? Amino acids are going to be the same way, proteins, right? When it comes to proteins, amino acids, generally they are going to get moved back into the blood as well. They're going to be reabsorbed completely back into the blood as well. Okay. Small and medium size can appear in the filtrate, like I said way back in the beginning, but generally it's going to be reabsorbed. When would it not be reabsorbed? Again, under high amount of stress, running excessively. If you have an excessively high protein consumption, greater than probably 2 or 3 grams per kilogram body weight would be probably up in that realm. So there's no evidence to suggest that a high protein diet would be an excessive amount of wear and tear on the kidneys and cause kidney breakdown. There's actually a lot of evidence to suggest that that doesn't happen at all. Excuse me. So just because you have a very high protein consumption doesn't equate it to kidney damage under normal circumstances, unless you're an individual who has only one kidney, who has some kind of kidney disease. In that case, you're not considered normal in that regard. So potentially high protein consumption can affect the kidneys, but there's a lot of new evidence to suggest that high protein consumption doesn't do kidney damage like we once thought. And that's a relatively new thought. The thought that high protein diet would destroy kidneys, would destroy the nephrons, the glomerulus and whatnot, or cause excessive wear and tear on the glomerulus. That's an old school thought. That's like 1940, 1950 research that really hasn't been challenged until the last handful of years. So kind of interesting there, and how we look at how kidneys function and diet and what that all means. Even for somebody with kidney disease, they're seeing some adaptations that the body does when it comes to protein consumption, and it's not excessively a huge amount of wear and tear that we once thought, even with somebody who has some kind of kidney dysfunction. And there's only been one paper associated with that. So, that's why I suggest normal kidney people, not somebody who has a disorder. So when it comes to the nutrients, glucose and proteins, guys, it's generally 100% reabsorption. Okay. It is generally 100% reabsorption. There are a number of ions that are going to be reabsorbed as well. And the vast majority of this reabsorption is always going to occur within the proximal convoluted tubule. Okay. I know I did not ask the percentage, but I know the vast majority of reabsorption occurs within the proximal convoluted tubule just because of the sheer anatomy of it, the microvilli. When we look at sodium reabsorption, because I'm going to ask about sodium and water on this exam, there'll be some questions, multiple choice wise, talking about how they get reabsorbed, where that reabsorption occurs, predominantly, and where I can also affect it. There are a handful of questions according to those guys. There's a number of other different ions that I mentioned and talking about, but there's only a few that I'm talking about. There's only a few that I will ask questions on the exam. That's what I'm trying to spit out here. Okay. Sodium is one of those. Okay. You should know where the vast majority of sodium reabsorption occurs, in the proximal convoluted tubule, like I keep saying. Like I said that a third time. Aldosterone can affect sodium reabsorption. When I increase sodium reabsorption, I also that's an O. I also increase my reabsorption of water. And we're going to see, I'll just go over here real quick, okay? A huge amount of water reabsorption occurs in the proximal convoluted tubule. So who's doing all this reabsorption? Primarily the proximal convoluted tubule is, okay? Whether it's sodium, whether it's water, the vast majority of this reabsorption occurs right here. When it comes to aldosterone secretion, how do we increase that reabsorption of sodium? Well, all we do is increase the number of proteins channels, okay, within that principal cell. And I can increase my ability to reabsorb it. I have all those microvilli. I can put all of these there and all those higher number of microvilli. So I can have a large number of these sodium channels. I can have a large number of sodium potassium pumps as well. I can embed these into all those microvilli, and I can increase my reabsorption of sodium. With sodium, water always follows. With sodium, water always follows. Where do we find all these ions? In water. So water is always going to follow that. So if that blood volume drops, okay, I can increase that blood volume by increasing my sodium reabsorption. ANP does the exact opposite. ANP inhibits sodium reabsorption. And remember, reabsorption is getting back into the body. What did ANP do earlier? I have to increase my GFR. There was too much blood volume. So I want to get rid of more urine, get rid of more water. By doing that, I increase glomerular filtration rate. So we had increased GFR occurring there within the renal corpuscle. And ANP is also affecting the proximal convoluted tubule to inhibit reabsorption of sodium. If sodium doesn't get reabsorbed, it stays in the tubular structures. If sodium doesn't get reabsorbed, water doesn't get reabsorbed. And I can create more urine. Get rid of more water, bring down blood volume, help to regulate blood pressure. I'm not asking numbers on the exam in terms of where the percentage of where sodium reabsorption occurs. Just the vast majority of it occurs within the proximal convoluted tubule. That's all I'm going to ask if I was to ask that. Okay. Where sodium goes, water goes. And you can see right here, the vast majority of that reabsorption occurs there within that proximal convoluted tubule. Okay. Again, there's the amount of filtrate formed and ultimately the amount of that gets reabsorbed. I did not ask those numbers. At least I'm pretty damn sure I did not ask numbers in terms of these things. Okay. But I just find it amazing that we form 180 liters of filtrate and then reabsorb like 178 of it. It's crazy. But it is necessary. Why is it necessary? Because it's going to create a number of concentration gradients, okay, throughout this nephron structure. Okay. If you have a high amount of stuff within the PCT, then and low amount of stuff within that paratubular capillary around it, it creates a huge concentration gradient, okay, which allows us to now get it to be reabsorbed. And then we can fine tune it because, I mean, there's still a little bit of reabsorption that occurs down here within the nephron loop. There's still a little bit of reabsorption that occurs over here in the distal convoluted and the collecting duct. So there's still a little bit that we can reabsorb. But here it freely happens, easily happens because of that huge concentration gradient. We talked about it with the macro or with carbohydrates and amino acids, proteins. 100% gets reabsorbed in a healthy individual. Damn near the same amount gets reabsorbed here for sodium, guys. You reabsorb the vast majority of sodium as well. It may not be 100%, it could be 98%. Okay. But under normal circumstances, we can still reabsorb a whole hell of a lot of it right here. And then as you can kind of see here, we can fine tune it here, which we will, and we'll fine tune it here, which we will. Who's the water transporter? Aquaporins are the water transporters. Aldosterone can affect sodium channels, increasing the number of sodium channels, therefore increasing the sodium reabsorption. Water reabsorption occurs via aquaporins. Okay. And we generally have a consistent number of aquaporins always found within that proximal convoluted tubule. Okay. This water that gets reabsorbed here in this proximal convoluted tubule is referred to as the obligatory water reabsorption. And it has a certain amount, and it's the certain amount of water that always gets reabsorbed back into the body. Over here on the back end of the nephron structure, in the distal convoluted tubule in the collecting duct, I can fine tune it. I can fine tune it. And that's referred to as facilitative. Okay. This is where antidiuretic hormone can affect it. A&P can have some effectiveness to it over here as well. But ADH, antidiuretic hormone, it can affect the distal convoluted tubule and the collecting duct by varying the amount of water reabsorption that can occur here. You'll notice that there's no percentage here. Okay, there's no percentage here. Because this over here is where we fine tune the amount of water that we get rid of, which is to say amount of urine we ultimately can form. Okay. If you are sweating a lot, not drinking enough water, you're going to put a hell of a lot more aquaporins in there to get reabsorption of water to occur. If you're drinking a copious amount of water, then you're going to insert fewer aquaporins in the distal convoluted tubule and the collecting duct. And you're going to form more urine. Okay. Because we are helping to regulate that blood volume. We are helping to regulate the amount of water in blood. And here's these little bitty aquaporins here. Okay. And you can see water's going through them. And water's entering into the cell and then leaving the cell and entering into the back into the capillaries here. If a diuretic makes you go pee, an antidiuretic makes you not go pee. So antidiuretic hormone puts more of those aquaporins in the membranes. This increases the ability to reabsorb water. Antidiuretic hormone has its greatest effect on the I don't really have it here. Has its greatest effect on the distal convoluted tubule and the collecting duct. Aldosterone affects it in the proximal. Here at the very tail end of the nephron, we can affect water reabsorption via inserting those aquaporins back here on the collecting duct or the distal convoluted tubule via this antidiuretic hormone. Here at the back end of the nephron structure, we are fine-tuning the amount of water we are ultimately keeping in the body. And this is that facultative water reabsorption that I talked about, made reference to just earlier there. These I left off. Understanding sodium in water is most important when it comes to understanding reabsorption here. I didn't get into all the mechanisms involved with potassium or calcium or phosphates. If I do have one question on there, it'll be for bonus. So don't worry about it. Bicarbonate is important because of what we talked about in terms of transportation of oxygen and carbon dioxide. In terms of the transportation of carbon dioxide in the blood, remember we have carbon dioxide that can be bound to the portion around the iron molecule, the globulin portion. But the vast majority of carbon dioxide gets converted into bicarbonate ion. 70% of it. So this is going to be within the fluid component. So obviously that's going to be a component of the filtrate. As you can kind of see there, reabsorbing this is pretty important. Okay, so honestly, really the vast majority of it does get reabsorbed, not to the degree of 100% like glucose or amino acids or even sodium for that matter. But we do reclaim a whole hell of a lot of it. Okay, and even though it's not 100%, 90%, 80%, that is a lot. Okay, that is a lot. The reason why it's important to remember bicarbonate ions because If we don't reabsorb it, blood can become way too acidic. Okay? It can become way too acidic. So the body will have to replace this. And it can only replace that with increasing the delivery or the transportation of oxygen-rich blood to the tissues. So we're going to have to pick up our rate and depth of breathing, which would change the amount of gas exchange occurring within the lungs and will ultimately affect the pH of the blood as well. So getting this bicarbonate ion back into the blood is going to be something that's important to do because it is a major regulator. It is a major reservoir to help to regulate the pH of blood within the body. So bicarbonate ions, because they are found within the plasma portion of blood, easily get pushed out into the filtrate. The body wants to keep that bicarbonate ion. The body wants to keep that bicarbonate ion to act as a regulator for pH balance within the body. So we reclaim, we reabsorb the vast majority of that. You can kind of see we can affect a little bit of it within the nephron loop, but the vast majority of reabsorption always occurs in the proximal convoluted tubule. Okay. What if it stays within the tubular network? Well, all that means is we have now a pretty we're going to have a greatly affected pH of the urine as well. Okay. If we reabsorb greater than 90%, you're going to affect the pH of the blood. If we don't absorb, or if we reabsorb less than the 80%, that can affect the pH balance of the urine. So keeping it within that 90% reabsorption, 80 to 90% reabsorption rate is significant because it can affect the pH balance of the blood or the urine, respectively. There are a number of steps associated with regulation of that. I just want you to know why it's important. Why is it important to reclaim, to reabsorb all that bicarbonate ion? And that is to not affect the pH of the blood. Okay? To not cause it to be too acidic. Too acidic, oxygen won't bind to red blood cells, and that's going to create a whole host of problems here. The last major thing that I would really like you to remember, because I know I asked a few multiple choice type questions on the exam about it. Is urea. Urea is unique. Whenever you think of urea, you think of pee, urine. It's the bad stuff. In the grand scheme of things, you only get rid of about 50% of urea. Urea helps to form this concentration gradient that always exists inside the medulla of the kidney. It's this concentration gradient that is allowing the nephron loop to do what it do. This ability to reabsorb and hold on to urea is important because it helps in what the nephron loop does. And the nephron loop is involved with reclaiming some bicarbonate ions, reclaiming some potassium, reclaiming water, reclaiming sodium. That urea stays down in here in a very high amount because we pump it out. And that urea stays here in the tissue deep in the renal medulla. It is the presence of that urea that allows a concentration gradient to exist down in here. Okay? If we absorb the vast majority of it, 65% of it, up here, it's not going to be very easy for it to freely go from in here to in here unless we've created a concentration gradient. And that's what urea does. Urea allows us to create this concentration gradient deep within the renal medulla to allow the nephron loop to fine-tune our ability to get rid of or hold on to sodium, water, bicarbonate ions. Okay. So the nephron loop can only function if we have a concentration gradient that is created. And that's what urea does. Okay. Again, I didn't ask the number, the percentage of it excreted, but you can see when you go pee and you get rid of that urine, only 50% of it is truly urea. Okay. You hold on to a whole heck of a lot of it. And you hold on to a whole heck of a lot of it to establish a concentration gradient deep in the renal medulla. Uric acid, I don't believe I asked about. It's kind of in the same realm with urea. Creatine, I didn't talk about. It's involved in protein breakdown within muscles. I didn't get into it. Okay, urea is the last of the components that I ask about what happens to it. Why is it important? Okay. Why is it important? When it comes to other substances that are secreted, there are a bunch of other stuff. Secretion, again, is the only thing that happens with secretion is getting it from the tubes, from the vasculature and secreting it back into the tubular structures. All right. When it comes to getting rid of certain substances, secretion is how it occurs. Drug metabolites are secreted. There are some examples. I didn't ask for examples. A number of metabolic waste are secreted. Okay. They're going to get pushed into the filtrate. They're going to be reabsorbed, and then they're going to be ultimately secreted again. A number of various different hormones can be urinated out as well. Okay. HCG, right? Human chorionic gonadotrophin. That's what you pee onto the stick to figure out if you're pregnant or not. Okay. The body secretes that. It doesn't need it. It's waste. Okay. If aspirin's been filtered out, it's waste. The body didn't have a need for it. Penicillin, sulfa drugs, all those things can be actively secreted. So in the terms of urine formation, all we've talked about is the glomerulus and the proximal convoluted tubule. That's it. That's where the vast majority of stuff is happening. We just fine tune it along the rest of the way. Nephron loop, distal, collecting duct. It's there in that distal convoluted tube in the collecting duct that ultimately we fine tune it. There's a little bit reabsorption, but not a whole hell of a lot. There's secretion. And mainly we talked about secretion in terms of water. So secretion is just a fine-tuning phenomena. That's it. Filtrate got it all out of the blood and into the tubes. Waste. Well, the body's like, crap, I need that. I'm going to reabsorb it. How much? I need all of it back. And then we fine-tune it through the nephron loop, through the distal convoluted tubule, through the collecting duct. And once we've reached that renal papillae, it's pee, it's urine, it's gone. And we don't cause any more secretion. We don't have any more reabsorption occurring at all. So slide 60, I didn't really ask any questions about it. The last few slides, especially these two, this is where we do see reabsorption and secretion. Okay. The nephron loop is that bend. Okay. Is that bend. That nephron loop has blood vessels that are closely near it. That's the vasa recta. The blue is the nephron loop. This nephron loop extends deep into the renal medulla. It's the nephron loop of primarily these juxtaglomerular nephrons, or juxtamedullary nephrons, that are going to help us to establish this concentration gradient that exists within the renal medulla. What do we have floating around down here in that renal medulla? Urea. Okay. It's that reabsorption, okay, or I'm sorry, that secretion of reabsorption that occurs, or that secretion of urea into the tissue that occurs in the renal medulla, that forms a concentration gradient down here with this nephron loop. This interaction between that high concentration gradient of the tissue and the contents of the tubular structure in the nephron loop is referred to as the countercurrent multiplier system. Okay. And that's right here. Okay. As we descend deep into the medulla, we come into contact with more and more urea. That creates a higher concentration. And the numbers are here. The numbers are not important. The numbers help you to visualize it. I do not ask the numbers on the exam. Okay. The concentration of the fluid right here at the very top of this nephron loop, so in the proximal convoluted tubule, is referred to as 300 milliosmoles. That's what MOSM stands for, milliosmoles. Again, I didn't ask about it on the exam. As we descend down this descending nephron loop, okay, it gets deeper and deeper into the renal medulla. As you get deeper into the renal medulla, it becomes more concentrated because of urea. Everything likes to be at equilibrium in the body, and this region is no different than any other region. Okay? So to create a isoosmotic condition where inside the tube is the same concentration as the surrounding tissue, as this fluid descends, the only way to make the inside more concentrated is to have it be permeable to water. So in this descending limb of the nephron loop, water is leaving the tube. As water leaves the tube, sodium ions, chloride ions, bicarbonate ions, they stay. So those solutes stay inside as water leaves. That means the concentration of the tubular content gets greater and greater and greater. If all that water leaves, wouldn't that water down the medulla here? Sure, you'd think so. Well, that's where we have this. Okay, that's where we have this vasa recta. Okay, that vasa recta, right? Yep, right there. Okay, because as the contents of this tubular fluid move down, it gets more and more concentrated. That's what this is. Okay, this countercurrent multiplier system. It gets greater and greater in concentration. It multiplies. As that tubular content gets down here towards the bottom of the bend of the nephron loop, that's it at its greatest concentration. As it comes back up, it's going from high concentration to lower and lower and lower concentration. Here's where we fine tune it. This ascending limb of the nephron loop is permeable to salt, NaCl. As sodium and chloride ions leave the tubular fluid, it becomes less and less concentrated. Again, if all of that salt left the tubular structure, wouldn't that affect the concentration gradient in the surrounding tissue? Yes. But that's where we have the vasa recta. Okay. The vasa recta are here to help maintain this concentration gradient. Okay. So here's my descending limb. So my descending limb is permeable to water. Okay. Tubular fluid becomes more concentrated as it descends, establishing a concentration gradient right here, right? Ascending limb, salt's being pumped out. It's impermeable to water. Only salt can leave. Again, staying isoosmotic with the surrounding tissue. But I've created this concentration gradient here because of that urea that allows this water to be pumped out in one and salts to be pumped out on the other side of the loop. And I can have a little bit of reabsorption occurring here of water and a little bit of reabsorption of sodium occurring in this nephron loop because of that urea presence from earlier. All that water leaving the descending limb of the nephron loop. If all that water was leaving the descending limb of the nephron loop, entering into the tissue, you would think that it would ultimately water it down. and it would. But we don't keep it there. The vasa recta that are mirroring the travels of the nephron loop help to establish what's called a countercurrent exchange system. They pick up what the nephron loop gets rid of. So as this descending limb of the nephron loop is getting rid of that water and becoming more and more concentrated as it comes down, we pick it up. This picks up the water, okay? So it leaves here and gets picked up into here. As salt is being pumped out here, it gets picked up here. This helps this blood as it's traveling down. Again, it's countercurrent. Okay. This becomes isoosmotic with the surrounding tissue, and it maintains that osmolarity with the surrounding tissue all the way around the bend in here. Concentrated here, why? Because it's been picking up all this dang salt that this ascending limb of the nephron loop has been giving off. And it becomes more and more concentrated as it heads down. This is picking up all of that water. And it helps it to maintain the osmolarity of the fluid content of blood with the surrounding tissue. Okay. Why is this important? Because we can fine tune a little bit more reabsorption and secretion here within that nephron loop. How did we do that? With reabsorbing, okay, or recycling that urea. We only get rid of 50% of the urea every time. 50% of it leaves the collecting duct and heads into the interstitium, into the tissue of the renal medulla. And I can fine tune how much water stays inside here, how much salt stays in here. I can fine tune that a little bit, okay, by establishing that concentration gradient. And what have I maintained the entire time? I've maintained the proper osmolarity, the proper pH balance of blood in the process. Okay. But I fine-tuned my water. I fine-tuned my salt a little bit here with reabsorption and secretion here within that nephron loop. And then I can manipulate it at the very end, right? In the distal collecting tube, distal convoluted tubule, and the collecting duct. I can fine tune. There's a tiny bit of reabsorption that occurs there, but not a whole hell of a lot. Okay. I can fine tune it by secreting a little bit more, right? Through hormonal influence on that distal convoluted tubule and the collecting duct. I ask a handful of multiple choice questions. Okay. What's a handful? Multiple choice questions into who's moving where, the descending limb of the nephron loop, what's actively being moved out. Water. In the ascending limb of the nephron loop, what's actively being removed from it? Salt. Why? To maintain the concentration gradients of the contents of these tubes with the surrounding tissue. The countercurrent multiplier system involves the nephron loop. The countercurrent exchange system involves the vasa recta. Tiny little blood vessels that surround the nephron loop. Who is this primarily occurring in? This is really only occurring, and by only I mean countercurrent systems here, are only occurring in juxtamedullary nephrons. They're the only nephrons that have nephron loops running deep into the renal medulla, helping us to establish a concentration gradient and allowing the nephron loops to fine-tune reabsorption or secretion of sodium, water, chloride ions, bicarbonate ions, anything that we've discussed. Distal convoluted tubule and collecting duct, their claim to fame when it comes to urine formation is being manipulated by hormones. Facultative water reabsorption is affected by hormones that affect sodium and water on the distal convoluted tubule or on the collecting duct. And hormones are going to be secreted not because of what the kidneys tell them to do, but because of what the system is telling them to do. This is extrinsic control. Am I sweating a lot? Am I outside exercising on a really hot day, not drinking? Okay, I'm sweating. I'm losing a hell of a lot of water. I'm losing a hell of a lot of sodium in that water as well. So it's just causing more and more water to go out. How can I regulate blood pressure in the body, trick the brain, and affect the kidneys all at the same time? Hormones. Antidiuretic hormone, renin, angiotensinogen 2, all that fun stuff. Okay. They make a great number of multiple choice questions and going from one point to another, how one affects the other. Again, this just reinforces that urea concept that I talked about earlier. Okay. Helps the concentrating process. Yeah. It's just cycle between the nephron loop and the collecting duct. Okay. Okay. Again, I didn't ask the percentages. It's just there to give you a frame of reference. Okay. When it comes to it, most everything when it comes to glucose and amino acids are always going to be reabsorbed. Okay. Sodium is damn near 100%, so I just put it there with it. Bicarbonate ions, roughly between 80 and 90%. Again, that's to maintain proper pH balance of blood and urine. Water is going to be regulated. Okay. There's 65% that pretty much does get reabsorbed all the time, but the rest is tightly regulated by antidiuretic hormone, aldosterone, and the situation. Are you outside on a nice hot day, sweating and not drinking water? Okay, well, now renin gets secreted, right? Vasoconstricts because you're losing blood volume, because you're losing water. Helps to raise the blood pressure so you don't pass out. Triggers the thirst centers for you to start drinking water. And it causes angiotensinogen 2 to do what it do to the kidneys to help decrease GFR. Constricting of those mesangial cells and limiting GFR.